So what we're going to be doing this week is we are going to be having our introduction into running reactions, and this is something that chemists do all the time, whether it is converting one functional group into another, for example a carboxylic acid into an alcohol, or we might do a reaction to make a new carbon-carbon bond, like this one. So whatever kind of reaction we're doing, one of the key things that we need to figure out is how do we know when this reaction is done? So we need a way to look inside the flask and be able to tell when the reaction is done. Well, before we can do that, we need a reaction that we're actually going to do. And the reaction that we're going to be doing this week is called oxidation of an alcohol. So what we're going to be doing is we're going to be taking a compound called 9-hydroxyfluorine, and we will be oxidizing it to form the ketone fluorinone. A few interesting things about this reaction. One is that it's going to, when we oxidize, we're using an oxidizing agent called chromium trioxide, and there are many other oxidizing agents for alcohol. Some that you commonly see in the lab are shown here. One of the things that many of, but not all, of the oxidizing agents that we're using have in common is that they have transition metals in them, and chromium containing reagents are very common. This particular chromium reagent is bound to a resin. It's basically a plastic, and it's very nice to have the material bound to the resin, and I'll talk about the oxidizing material bound to the resin, and I'll talk about why that is in a second. So here's the setup for our reaction, and this is called a reflux reaction, and you should have watched the video from MIT. And the setup that we're going to be using is very similar to the MIT video. Pretty much the only two changes is we're not going to put any grease on the joint between the reflux condenser and the round bottom flask. The reason for that is that we find that grease is not generally needed unless you're heating up something that's very basic that could etch the glass and cause these the joint to stick together. Otherwise it just becomes an impurity in your reaction. The other difference is we're going to drop in all these reagents all at once and then put the reflux condenser on. Now just a quick note about the reagents that you're going to have in here. First off, the chromated resin, you are going to use 1 gram of that. Second, the alcohol, if you look in the handout, it says you're going to be using 0.55 millimole. Okay, and you'll also be using 3.5 milliliters of toluene, and finally 1 stir bar. Now the important thing to note here is that this number is not in grams. So you're going to need to calculate the number of grams of alcohol you're going to need to be using, and you need to have that information with you when you come into lab. Just a quick little bit of information here about the polymer supported reagents, and that's what we mean when we say this resin that we're using. And the oxidizing agent, in this case, remember it's CrO3, it's bound to that resin, and so that resin is porous, and as you run the reaction the resin is not going to dissolve, but the alcohol can diffuse into that resin, and then once the alcohol that we're using has diffused into the resin, then the chemical reaction, which is an oxidation, can occur. And then that, remember that we're going from an alcohol to a ketone, and that reaction is called an oxidation, and we'll see a whole lot more of these as we go further on in the semester. Once that carbonyl compound is formed, then it's going to diffuse out of the resin, and it'll be in solution. And then the nice thing is in order to remove the chromium trioxide, then all we need to do is filter out the resin, and all the chromium comes with it, and what we're left with is our ketone product, and that ketone product is going to be dissolved in the solvent, which you recall is toluene. Also recall that in order to do the filtration, we're going to be using a filter pipette rather than a filter and filter paper, and that's because this reaction is on such a small scale, we would lose most of our product on the filter paper. So we want to be sure to keep the most of our product, so we're going to use something called a filter pipette, and I made a separate video on how to make a filter pipette, which you should watch. Okay, so that's how we're going to run the reaction. Now we got to think about how are we going to follow it, or how are we going to analyze it. And we're going to use the technique that we've used before, TLC, thin layer chromatography. And the first thing we need to think about is how are we going to know what the RF value is, approximately. We can't predict exactly an RF value. Remember, they're not necessarily reproducible 100%, but we can get a general idea of if the starting material or the product, what are their RF values going to be relative to each other. And the first thing that we need to look at is which of these compounds, the starting material or the product, is more polar. So the first thing to think about there is to think about the intermolecular forces. So I'll let you do that. Okay, so now that you've thought about that, I hope you've seen that the starting material has an alcohol in it, therefore it can undergo hydrogen bonding, whereas the product has this ketone functional group in it, and therefore it's just going to have dipole-dipole interactions. So that means the alcohol is going to be more polar, and that's important in terms of the RF value in the TLC. So why is the polarity important? Well, as we saw, the starting material is more polar, and because it's more polar, that means that it can stick to the TLC plate. And a material that sticks or absorbs more strongly to the TLC plate is going to have a lower RF value. So here is sort of a crossways image of a TLC plate, where this is the plate or aluminum backing, and this is the silica gel. And notice the silica gel has all these hydroxyl groups on it, and that is a very, very polar absorbance surface. So if we have a more polar molecule, it's going to be more strongly adsorbed onto the absorbent and is going to move more slowly. And the less polar molecule, in this case, is the product, and it's going to move faster or have a higher RF. And therefore separation occurs. So when you're following a reaction by TLC, you're going to start out and you're going to spot the reaction mixture and the starting material on the same plate at different time intervals. So here we have a TLC plate, and we've spotted the starting material because we always want to have a comparison, and we have also spotted the reaction mixture. So after we've taken the sample from the beginning of the reaction, so we've just mixed things together and are starting to stir them up, and this TLC plate has been developed, what we can see is in our reaction mixture we are starting to see a new spot, and that new spot is at a higher RF, and we talked about how the higher RF is the lower polarity compound, so we can assume that, or hopefully this is our product, and we can see also that we still have some starting material around. So this is telling us that, not unexpectedly, right after we've mixed all the reagents together, the reaction maybe is starting a little bit, but it hasn't gone to completion yet. So this is what it might look like after a few minutes of heating the reaction. And you can see here that we're starting to form. Remember, TLC is not quantitative, it's qualitative, but you might see that your product spot is getting a little bit bigger, and your hopefully that your starting material spot is getting a little fainter. And here's what it might look like after you've been refluxing for a few minutes. So this TLC plate here might be just after it was heating, but this TLC is after 5 minutes of actually refluxing. So the solution is boiling, it's going up into the condenser, it's dripping down, and so this is what it might look like. And so after you've been going along and checking your reaction every 5 or 10 minutes, eventually, one hopes, you will get to a TLC plate that looks like this, where, as you can see, you have absolutely no starting material and all product. And so after you do the happy dance to celebrate the fact that your reaction is complete, you are ready to work it up. And in this case, the workup is very, very simple. Pretty much all we have to do is filter the reaction solution using the filter pipette, and then that's step 1. And then step 2 is we're going to evaporate the solvent, and we'll let that happen over the period of the following week. It's only a few milliliters of solvent, and it should not be a problem. Alright, so here's your game plan to get ready to do this reaction this week. Just a note that you're doing the reaction individually, so each one of you will be running your own reaction. As always, you want to have a plan or an outline for what you're going to do when you come in. It's a good idea to set up some TLC plates. Go ahead and take 3 TLC plates and spot them with starting material, so they'll be ready to go. Then you can get your reaction going. And then as you're doing this, you can remove a sample you saw at the beginning. That's an example of that. After 5 minutes of heating, after 5 minutes of reflux, and then every 10 minutes after that if it is still not done after 5 minutes of reflux. And as I said, at completion, you're going to remove the resin via filtration through a filter pipette. Allow it to evaporate to remove the solvent. And the drying step that's the sodium sulfate that you're packing the filter pipette with. That is the drying step. Just a reminder about some of the important things you need to have in your PreLab. You should have the reaction. You should know the amounts of all the reagents you're going to be using, including the starting material, in grams. Okay. You need to have pertinent information about reagents, melting points for things that are solids, boiling points for things that are liquids, like the toluene solvent you're going to be using. That's very important. Hazards, a reference, even if it's just to a web page. You need to have calculated the theoretical yield, and finally you need to be sure that you have a plan for what you're going to be doing, a plan or an outline of what you're going to be doing. And that's it. We'll see you in lab.